Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Combined Science Biology Paper 1 Higher Tier from 2024. Question number 2 is on the topic of cell biology. A student investigated the concentration of salt in solution Z. The student used a method involving osmosis, which means this is a required practical that you will be being tested on. The student used tubing made of partially permeable membrane, which is to stimulate or actually represent a cell membrane. This is the method used. Cut 6 pieces of tubing to the same length. Tie one end of each piece of tubing. Put the same volume of solution Z into each piece of tubing. Tie the other end of each piece of tubing to form a sealed tube. Record the mass of each tube. So the same volume is in each piece of tubing. Record the mass of each tube. Place each tube in a different concentration of salt solution. After 2 hours, remove each tube from the salt solutions. Record the mass of each tube. Figure 1 shows one of the sealed tubes in a salt solution. So you have the salt solution in the beaker solution Z, which also has salt inside it. We don't really know the concentration of it. The partially permeable membrane. Now the tube looks like a firm sausage, which means water should have actually moved in. What was the independent variable for the investigation? The independent variable is the concentration of salt solution that was used. The change in mass is the dependent variable. The time is the control variable. Volume is also the control variable. The student dried the outside of each tube with a paper towel before recording the mass. Why is it important to dry the tubes? Well, the water on the tube would affect or increase the mass. We are only interested in the mass of the water that is inside the tube, not outside. Table 1 shows the results. So at 0, at the start, this is the mass change. They haven't worked out the percentage change here, which means we will have to do it in the next step. Yes. Let's have a look at the concentrations. So from 0.2, 0.4, it's going up by 0.2 all the way up to 1 mole per dm cube. And it looks like the mass is actually increased in all of them. Mass increased here. The last two, the mass decreased compared to the start. That means here the reason for the mass to increase, water moved in. Here the reason for the mass to decrease is water moved out of the tube. Right. So calculate the mean value x in table 1. Give your answer to 1 decimal place. So the percentage change is the percentage change in mass equals this 0.96 divided by the starting mass, which is 15.54, and multiplied by 100 to get it as a percentage. That would give you 6.1776. So in one decimal place, it will be just 6.2. Complete figure 2. You should plot the percentage change in mass from table 1 for salt concentrations of only 0.2 to 1. So you don't have to plot the 0 that we actually calculated at the start. Draw a line of best fit. One of the results has been plotted for you. So here's the graph as a whole. So you can go up to the negative values. They have done 0.8. Let's do, while we are here, what the percentage change in mass for 1 is minus 2.5. So if that's 2, this is 2.5. So that's minus 2.5. Let's go up. So at 0.2, it's 4.1. Now that's 10 boxes, so each box is 0.1. So that's easy. So 4 and 1 box up, and 2.3, 2 and 3 boxes up, 1, 2, 3 boxes, and at 0.6 is 0.5. 0.6 is 0.5, 1, 2, that's 0.5, isn't it? Yeah. Okay. So let me bring that into focus. So you can see all the points now, and they're not even a curve, they're actually a straight line. So I could draw a straight line, can't I? So let's draw that. Oh, I meant it to go straight. There you go. Now it looks like 0.1 is an anomaly because it's falling out of the line, because the rest of it are all falling in a straight line. Determine the concentration of salt in solution Z. Now that's when this line actually crosses the 0 value. Now why is this point? Let me work out the value first. So this is 0.616263, so that's 0.63. At this point, the overall or the net change in mass is 0. That's what it's saying. So when the mass of the solution, the concentration of the salt solution outside and inside is equal, then the overall exchange of water is going to be the same. So this would be 0. What did we say? 0.64, was it? 63. Let me double check that value again. Yeah, it is definitely 0.63. Now the marking scheme says value from students line of best fit. So as long as we've got our value correct from this line, it's fine. If you haven't drawn a line and if you guessed it, your value should be somewhere around 0.64 to 0.66. That actually brings us to the end of this question. Thanks for watching, and I will see you in question number 3.